That first slide gives you an overview of what selective transport is, right? So the whole thing about transporting is you're transporting substances across the cell membrane. You also are transporting using transport proteins as well. And of course, like we mentioned, there are two mechanisms of transporting. One is through passive transport, right? The other one is through active transport. We said that active transport requires what? ATP, correct? And we said that passive transport, it requires what? No ATP. We got that, right? So that's what we went last class. Now, on the passive transport, okay, we had a diffusion, osmosis, and facilitated diffusion. We said that diffusion was?
Diffusion was high to low.
High to low what?
Solid.
So is the diffusion, it was the movement of solutes or water? Which one was it?
Solid.
Solutes, from a high concentration to a low concentration. So it's solutes from high to low concentration. And remember, this is a semi-permeable membrane, meaning the membrane is permeable to the solutes. Now, osmosis was the movement of what?
Water.
Water. And then that is, of course, water move what? From a what to what?
Low.
Is the opposite of the opposite of this. So from a low concentration of solutes, right, to a high concentration of solutes. Again, we're saying that the membrane is permeable to water. If it be impermeable to water, then there's no way that water can pass through. So far, so good. We also said, we talked about this, and we said that the plasma membrane or the cell membrane was semi-permeable, right? Some things are permeable to it, others are not. The other one that we talked about, it was the facilitated diffusion, correct? And facilitated diffusion was about what? Uses a protein, a transport protein. These transport proteins were known as the integral proteins. You guys remember? The proteins that went across the whole membrane. So these transport proteins are going to have create either channels or are going to be what? Carriers. If you create a channel, it's just a channel that is open, correct? If you have a carrier, that means it's most of the time is going to carry a specific ion, right? So that means that the ion is going to bind to that carrier. Okay, so you're carrying to a specific ion. So far, so good? Okay. So the last thing that we said last class, it was the osmotic, which is the movement of water. And we were talking about the cell, of course. You had either an isotonic, a hypertonic, or a hypotonic. When they refer to this, they're referring to the solution. And the solution is what is outside the cell. So the cell's in the middle. An isotonic means that you have the same components or the same amount of solutes inside and outside. So there is a net movement of water to be what? Out or in? What is the net movement of water in the isotonic solution? Iso means what, guys? So what's the net movement? Equal. Same inside and outside. Hypertonic solution, we said that you have more solutes outside and less solutes inside. That means where's the water going to move? In or out of the cell? It moves where the more solutes are. Okay? That is called what? Guys, when the water moves from inside of the cell to the outside of the cell, what do we call it? In this case, it's osmosis, correct? So this is known as hypertonic, right? And this one, it's going out of the cell. Then the last one was the hypotonic. The hypotonic one, it was you have low solutes on the outside and more solutes on the inside. That means that the water overall movement, it's going to be where? In or out? So the net movement of water will be in to the cell, right? Or in the cell. So far, so good? Everybody got that? Now, a hypertonic solution, where do we see that in real life? And that's most likely what? Rivers, right? The regular river waters. Where do we see hypertonic solution? As the seawater, all right, salt water. You guys get it? So normally, if somebody dies, so we're going to go into CSI, right? If someone dies and you find their body, okay, and you see that the cells of this dead individual, okay, are shrinked, where did the person die? If the cells are shrinked, where did the person die? In the ocean. Why? Because the water is moving out of the cell. Does that make sense? So it means the person dies somewhere in the hypertonic solution, right? Say, most likely the salt water, especially here in Florida. Now, if you found the body in the ocean, and when you take a piece of the tissue and you look it under the microscope, the cells are swollen. When you compare it to other tissues, what does that tell you? The person died where? You found the guy in the ocean, but the cells are swollen. Where did the guy that was dead? On a river, even though they dumped it on the ocean just to look like the guy was, I don't know, in an accident or whatever in the sea. Does that make sense? But the cells would not lie. Is that clear? Okay. So that's what we use these things. Okay. Now, the next stuff, we just went over this one. The next one will be the active transport. So the most common part of the active transport, again, is that it uses ATP. The number one example for active transport will be what? It will be your sodium-potassium pump. The sodium and potassium pump, and by the way, these are two positive. The sodium-potassium pump, it pumps sodium and potassium in and out of the membrane. Please understand that when we spoke about the cell, in the cell itself, the inside of a cell is negative due to the high amounts of proteins. So it's due to high protein content. Okay? The outside of the cell is overall positive. What is the overall ion that's causing this overall positive charge on the outside of the cell? And that ion is known as sodium. Okay? What is the main ion inside the cell? Potassium. But please note that both sodium and potassium are what? Positive charges. So the charge inside is overall negative and is due to the protein content in these organelles. Yes? So as you move sodium into the cell, as sodium opens, right, the sodium potassium pump, as sodium moves into the cell, what's going to happen to the overall negative charge? If you put in more positive into a negative, what do you end up getting? If you have two negative charges and I put 15 positive charges, what do you have? Positive, right? If you have -$2 and I give you $5, what do you have? Positive 3, right? So as sodium comes inside the cell, right, through this active transport, now sodium is going to stay in here and sodium is going to change the charge from negative to positive, overall more positive inside the cell. Now sodium is leaving the outside, correct? What's going to happen to the outside positive charge? It's going to become more negative. Does that make sense? So what have we created? We changed the polarity of the cell, correct? So we call this depolarization. So depolarization is actually you're changing the poles of the cell. The inside was negative, the outside is positive. When you pump it, right, through the ATP use of the sodium potassium pump, then we call that depolarization. Now, do you want your cells to be all the time depolarized? Is that normal? No, this is normal, right? So what do you have to do then? You have to put your cell back to normal. What is normal? Then if sodium is inside, what is going to happen now to the sodium? Sodium needs to be pumped back outside, so the overall charge outside will be overall positive, and the inside will be now negative. So we change, so this part right here that is going back from depolarization, we're calling it this now repolarization. Everybody get it? Because you're going back. So that's the use of sodium potassium pump for most of the times. Now, normally after transport, the reason why we use ATP, because it goes against what? Concentration gradient. What is the concentration gradient? I believe is the opposite of what? When you go against concentration gradient, you're going opposite to what? To diffusion. Does that make sense? Because the solutes, you're now pumping against concentration gradient, hence you need ATP. So it's like you have 1,000 students in here, right? If I give you $1 million, will you consider coming inside the classroom? How many of you would do it for $1 million? What about 5? What about 10? All right? That's all the body's doing. It's using ATP to force things to go against concentration gradient. So all these pumps are using by what? The same protein pumps that we talked before, right? The integral proteins. Now, what goes against concentration gradient? And there are two processes in the cell that uses it. One is the endocytosis, and the other is exocytosis. We learned that we use exocytosis to put things outside the cell, both waste products and what else? Proteins, correct? Because as you assemble proteins on the rough endoplasmic reticulum, you're going to send them outside. Now, endocytosis, you have different type of endocytosis. In other words, it's a way that your cell uses the food that you ingested, and now your cell is acquiring the food, so it can be used to be converted to ATP. So that process of endocytosis is known as either phagocytosis, because endo is just bringing it inside, right? So the process can be phago when we use solids. What are some solids? I don't know. A simple glucose molecule, a simple amino acid, or a simple fatty acid. You guys get it? That's what's going to be used. So that's solids. What is pinocytosis? It's liquid, means cellular drinking. Most likely, what is in the liquid? Your ions. So there are ions in the liquid, the sodium, potassium, chloride, magnesium. That is the process of pinocytosis. So there are two processes that uses endocytosis. Hopefully that just a little more clear now, guys. Okay. Now, we talked about the pinocytosis. We talked about the phagocytosis. There is another one which uses a what? A receptor. Which one are the ones that uses a receptor? Really simple. If you want to select for a specific ion, let's say iron, because your cell requires iron, or magnesium, okay? The iron molecule is going to have a specific shape, the magnesium is going to have a specific shape. That means on the outside of a cell, you're going to have a shape that can accommodate for the iron, or you have another receptor that can accommodate for the magnesium. Once it binds to the receptor, now your cell is going to create something like this, right? Now bound already to the magnesium or bound already to the iron. You get it? So as the cell moves around here, it moves the cell membrane, then that's going to be, again, a process of endocytosis. What have we done so far? We have covered the whole chapter. The chapter is about these things. If you understood these things, that's a pretty much summary of what is rest to come on the PowerPoints. Okay? They're just going to go a little more in details, but that's as detailed as you may need. Okay? So let's go to the next things. So everything goes against what? Everything goes through what? Plasma membrane, correct? That plasma membrane is going to be regulated by, of course, the exchange with the environment. What environment are they referring to? Remember we talked about the solutions. We said that you had a cell and then you have a surrounding liquid, right? The solution. So this is the solution, right? That solution in the cell, in our body, is called what? Extracellular space. Everybody got that? And the inside of the cell is called what? Intracellular cellular space. Everybody got it? If you're lost, please let's stop me. So, passive transport, as we mentioned before, uses ATP, yes or no? No. So we have simple diffusion, channel-mediated diffusion, facilitated diffusion, and of course, osmosis. Which went over it? The next one is the active transport. Active transport versus passive transport is that it uses what? ATP, because it moves things against the concentration gradient, right? Against what we know as the proper diffusion. Yes? So that was what? A sodium-potassium pump. A active transport can be primary or secondary. It doesn't matter whether it's active, whether it's primary or secondary, is that it still uses ATP. And you have a vesicular transport and the process of that vesicular transport. The vesicular transport I was referring to is the transport vesicles. Remember the transport vesicles that move from the Golgi apparatus outside the cell and release the protein? Those are the vesicular ones. So you have the process of endocytosis, which we said it was what? Either receptor mediated, pinocytosis for liquids, and phagocytosis, correct? For solids. The process of exocytosis is getting rid of what? Waste products and the protein for export. So that was fair off. So there is a video on the PowerPoint that you can watch, right, which is about positive transport, which basically explains this, right? You get a concentration that goes from a high concentration to a low concentration, and it gets, of course, dissolved. We also talked about the membranes. So membranes, if they're impermeable, means that nothing can go through it. Our membrane, our cell membrane, or our plasma membrane is not impermeable. Is what? Is semi-permeable. It's not freely permeable, because freely permeable means that everything can go through it. Solutes, water, everything. Right? So, it is selective permeable. That's the one that our cell uses. Okay? So, things that can go through that cell membrane is your lipid soluble, because again, this portion of the cell right here is what? Made out of lipids, correct? Because that was made out of a phospholipid bilayer. Here's your phosphate, right? Your phospho, and these are your lipids. So the inside, the middle, is a hydrophobic. Okay? Now, what other things can go through? Well, small things like what? The small water solutes, okay? But basically, oxygen and water can go through here, okay? Mainly the oxygen. So far, so good? Okay. So any lipid-soluble hormones. Later on, we're going to talk about this in 2086, all right, in the next anatomy. So hormones that are made out of lipids, they go directly into the cell, and they affect the nucleus a lot faster. So any medication that you use, right, or any hormones that are used that are made out of lipids, you're going to have the effect right away, instead of ones that does not. So again, simple diffusion is used by what? Lipids. Channel-mediated diffusion is used by what? The water and some of those ions. Facilitated diffusion is that we talked about the molecule of glucose that needed what? Sodium. So a facilitated diffusion means that the protein that is here is going to use sodium to open the glucose. And that's what we talked about, the Gatorade, remember? That was the best way to hydrate. And this happens again in your GI tract, especially on your small intestine. We talked about this already, osmosis, which is the movement of water, osmolarity and tonicity. So the osmolarity, again, is the movement of water, right? The tonicity of solution is given by what? Hypertonic, hypotonic, or isotonic. That's what they're referring to. Tonicity means if I have a solution that it has this amount, And this in here, what is the tonicity of the solution? Come on, guys. Where do you have more solutes? Extracellular or intracellular? Sorry? Extracellular. So what is the tonicity of the solution? Guys, all I'm asking if it's hyper, iso, or hypotonic. Hyper. So the tonicity is hypertonic. So again, that's tonicity. Don't get that confused. It's not another word. It's either iso, hypo, or hypertonicity or tonic. Okay? And we talked about that. So you can look at here. We talked about here as well. So isotonic, there's not net movement of water. The water just equalizes between the inside and the outside. That's what you really want to have in the body. A hypertonic solution, the cell is going to lose all the water, so your overall cell will shrink, like we said, right, on salt water. And on the normal hypotonic solution, the cell would tend to swell up, and basically they can also do what? Burst out. The rupture, right? They can rupture. Active transport, let's go over it again. So active transport requires what? ATP. What is the main one? Sodium potassium pump. Okay, the sodium potassium exchange pump, if you see that word, just is the same sodium potassium pump. Okay, it is an exchange pump. Why? Because it's exchanging sodium for potassium. That uses a carrier protein, right? And of course is a carrier mediated transport. Why? Because it requires a protein to carry things around the membrane. Okay. Here you can see the sodium potassium pump, and the way that the pump works is that, of course, it's an integral protein, and then there are sodium, 2 of those in, and there's 2 potassiums in and 3 sodiums what? Out. You need to know how many goes in and how many comes out. Okay? So most of the time, you're doing that against concentration gradient, right? Where's the sodium most likely found? We already said that it's most likely on the outside, right? Outside is the extracellular space, yes? So why are we pumping the sodium out of the cell? So we can make the cell what? Negative, more negative, right? The intracellular space. So far, so good?
Which normally happens when there's an action potential, right?
We want to have this because after you send the electrical impulse across the membrane, you want to have negative inside and positive outside, because this is your resting state. If you don't have this, then basically your membrane, if it's opposite, it will be positive and negative, right? This, we call it depolarization. And that's when you send an electrical impulse. Okay? So you need that resting state for it to send the impulse later on. So sodium potassium pump, again, as you can see it here, normally the way that happens, again, is 3 sodiums out and 2 potassiums in. Again, make sure you know that. And of course, is a active transport, so it uses ATP. Vesicular transport, we have the endocytosis or exocytosis. Why do we call it vesicular? Because, guys, it creates a little, this is your cell, that's your nucleus, so it creates a vesicle that migrates to the cell surface, and then once it does it, it's just going to bind to the cell surface, and it's going to excrete the content here. Correct? That's a vesicular one. Because inside the vesicle, well, you have your things in there, right? The proteins or the waste products. So that is either by receptor mediated, if instead of coming out, you want things to go inside the cell. So if you want things to go inside the cell, right, that means coming in. So this is exocytosis. And this is what? endocytosis, right? Is coming in. So the vesicle then is going to carry things inside the cell, especially to the mitochondria to make ATP. Okay. So you can see here the receptor mediated is, I think, is a lot better than my pictures. So you can see here that it's binding to the receptors, right? These are the receptors. And as it binds to the receptors, it's gonna encave, right? The cell membrane onto a point that you have a vesicle now inside. The vesicle then is gonna fuse to your lysosomes, right? And your lysosomes are gonna break down the food particles. Basically, if it's a pinocytosis, then it's drinking, it's in the fluids with sodium, potassium, magnesium, your ions. If it's, what is the other one? We talked about two. Pinocytosis and if it's phagocytosis, then it will be a little different. That will be mostly for solids, right? And of course, there's this one, which is what? Receptor mediated. So there are three types of ways of doing the endocytosis, right? This is all checked here in steps, right? So you can look at it. So again, pinocytosis is cellular drinking. That, of course, is the fluid content. So whatever ions are in the extracellular space, so any ions that are extracellular space, then they're going to be able to, again, create a vesicle, and that vesicle is going to be able to be used by the cell. Okay? Phagocytosis, we said that it was what? Solids, right? So anything solid, not only the amino acids, which is your protein, your glucose, which is a monosaccharide, or a single fatty acid. It can also be what? A pathogen, right? A bacteria. And you guys know every time you get sick is because a bacteria, right, is trying to get inside your body, and guess what? They got in, because that's why you have now the symptoms. Same thing for a virus. Every time you got the flu, that virus is doing what? It's getting inside the cell. And your cell, it tells the cell to make more viruses. Same thing with the bacteria. It tells your cell to make more bacteria, or it destroys the cell itself. So once you have the symptoms, that means that bacteria or virus already infected your cells. So it uses a process of phagocytosis. So it's not only for the food, but as well as what? The pathogens, right? The bacteria. So exocytosis, again, is the process of taking things out of the cell, right? And we talked about the proteins for export that they are coming from the rough endoplasmic reticulum. Remember that? Rough endoplasmic reticulum or bound ribosomes. Hopefully you guys remember that. And then, of course, the waste products, right? So that's as well as waste. So, again, endocytosis, receptor mediated, pinocytosis, cellular drinking, and phagocytosis for solids, right? Including the food and the bacteria. You guys are understanding a little better now, right? And here, this table, what it does is just compares everything together. So please take a look over this one just for comparison. Okay, but we talked about most of these things already. All right, so this one, let me just compare this one really quick. So you have the active transport. You have also a secondary active transport. A secondary active transport or primary active transport all requires a what? A carrier protein, right? A secondary transport is a carrier and an ATP. So carrier, substrate, and ATP, carrier, substrate, and ATP. You see that both of them uses what? ATP, and the carrier, which is your protein, correct? That's what we said. Now, the active transport, the primary one is sodium, potassium, chloride, magnesium. So these are the ions. Now, the secondary active transport, the only difference is what? Glucose, amino acids, and iodine. All right, so what is glucose, amino acids, and all these things? Glucose, amino acids, iodine. Glucose, amino acids, what is that called? When your cell puts it inside. Endocytosis. Remember? But in this case, it can do it through a protein itself. Instead of getting a whole chunk of amino acids and glucose, it can just grab one glucose molecule, okay, or as needed. It doesn't need to take 15, it can just take in one, okay?